Hi guys, Mr. Hyatt here. This walkthrough we're talking about mutations, and just a note on mutations. Remember, we've covered mutations at the chromosomal level. We talked about deletions and insertions and translocations and inversions. So make sure you still are familiar with those terms and what those mean in terms of chromosomal changes. And I know you guys all know this, but just remember that when I say mutation, I'm talking about a change in the genetic makeup. So just keep in mind that negative connotation for mutation isn't always the case. Sometimes a mutation is positive, sometimes it's negative, sometimes it's neutral. We're mostly focused on point mutations in this unit, so we're talking about small scale changes, maybe 1 or 2 or maybe 3 nucleotides. If it's something bigger than that, then we're kind of looking at something like a chromosomal alteration. So like I said before, the effects could be none at all, could be silent, or it could lead to some pretty bad changes. So let's kind of dive in and look at the specific kinds of mutations. I think insertions and deletions are really common sense. I think you probably understand those. And you probably at this point understand a base pair substitution. So a substitution truly is a substitution. We're switching one nucleotide for another. Here's a picture example. Sickle cell anemia is is caused by a mutant version of hemoglobin. And you can see right here that the point mutation is just a substitution from a thymine to an adenine. So that translates to a different amino acid sequence, which if you think way back to our very first unit in August, we talked about how primary structure of a protein impacts quaternary structure and then function. So if we've got a different structure here, we're going to have a different function. Notice that it's not a major mutation. It doesn't change us to a different protein. It just makes the protein less effective. So there are two types of substitutions. There's actually a third type that I know you're all somewhat aware of. We'll get to in a second. But a missense mutation is still going to produce an amino acid, a peptide that's the same number of amino acids. So we've changed the words, but there's still the same number of words. A nonsense substitution is going to, the point mutation is going to cause a codon that would normally code for an amino acid to code for stop. So it's going to stop translation. Here's a picture kind of showing that. The wild type would be the original strand. Here you can see we've had a base pair substitution where we've got a U instead of a C. So that's what you may have called the wobble effect before. It codes for the same amino acid, so that's a silent mutation. A missense mutation, again, is going to cause a different amino acid to be coded. Instead of glycine, here we've got serine. And then a nonsense is going to code for stop. So we're going to get a truncated or a shortened version of that polypeptide. So what effect does the wobble effect have on missense? Basically, if the correct third base is changed, then the mutation is silent, doesn't really affect the overall protein, doesn't affect the function in any way. It is just truly a silent mutation. But they still slow down the speed of making a protein because some tRNAs are harder to find, like it says on the screen. Some of them, essentially what that means is we don't make as many of all of them. So some of them are more rare. Think of it as like a long-term storage, short-term storage kind of a thing. We're not going to just carry around all those tRNAs all the time. We're going to create some as needed. We're going to keep a few copies in our long-term storage. So some of those are going to be harder to find than others. Missense mutations, again, remember this is, we're still coding for the same length of peptide, but it has a different primary structure, so we've got a different amino acid. it kind of depends on where it is. If we've got a mutation in the active site, that's going to cause a major impact because those active sites are so specific that any change at all is going to mess with the function. I'm sorry. If it's in another part of the enzyme, maybe it doesn't have any effect. Something else that plays into this is what substitution are we making? If we're making a substitution between say aspartic acid and glutamic acid, that's two acids. Chemically, they're going to behave similarly. So that's going to produce a lesser effect than maybe switching from, I don't know, glutamine to glutamic acid or something along those lines. Missense Effect stops proteins, so that's going to lead proteins that don't work at all. Or occasionally we can get lucky and the protein's going to function on some level because, like it says on the screen, our mutation maybe happened at the very end of the polypeptide and didn't really mess with things in a big way. There are things called sense mutations. We can change a stop codon into a reading codon. Like it says here, it's going to give us longer polypeptides. Sometimes they're functional, sometimes they're not. There's something called heavy hemoglobin. I don't really want to get into it right here, but it's an example of one of these sense mutations where essentially we're making a protein longer than it naturally would be. Again, anytime you're changing the DNA, you're changing the chemical structure of the protein, then it's going to change the function of the protein. Insertions and deletions are going to be inserting or deleting 1, 2, 3 bases. Sometimes it's more than that, but again, that's more of a chromosomal change. But what we see are these frame shift mutations. So we talked about that in class. Again, we're just changing where our spaces are in our words. That's kind of the best way that I can explain it. Here we've got our DNA template across the top. We've got our 5' to 3' end. When we look at our 5' to 3' mRNA, mostly matches here, except our T's or U's. Excuse me. Here in the bottom, we've had a deletion. So now our frame has shifted. Where we had an even stop codon at the end, now we don't have that UAA anymore. So our reading frame has shifted. Like it says here, if we lose 3 nucleotides, that's really not a problem because we're just deleting one amino acid, a whole amino acid. We're not shifting everything downstream of that amino acid also. So maybe the protein can still function depending on, again, where it is. If it's in an active site, maybe not. If it's somewhere else in the enzyme, we're probably still okay. There's kind of an example of that. That's a 3 base pair deletion. So there are a couple of things that just kind of get tossed in here, kind of vocab. Mutagenesis is the process that causes mutations. So that's where the word mutagen comes from. That's kind of where that root lies. So mutagens are going to be things that cause DNA changes. We've talked a lot about radiation. 5-bromouracil is another. It's a chemical that causes DNA changes that can lead to cancer. You guys know about mutagens, so let's just move on. There are spontaneous mutations, like we've talked about. There are those random errors that happen during DNA replication. We talked about that in class, so hopefully those make sense. So this comment I felt was worth keeping in the presentation. Again, a lot like we talked with BPA and a lot like we talked about some of these hormone blockers, the things that are most damaging to us are the things that are really close to the things we already use. So if something is similar to our nitrogenous bases, our bodies, our polymerases, all of our mechanics are going to confuse that. These things aren't really conscious thinking beings. They're chemicals. They're molecules that are interacting. So those molecules are going to make mistakes. They can be tricked. So if something mimics a base that we naturally use, our body's going to incorporate that. And that's going to lead to genetic changes. That's going to lead to all kinds of problems. So that's really the DNA specific mutations down at the DNA RNA level. I hope this stuff's starting to make sense.